Good morning, everyone. This presentation is the part of Dissolution Research Presentation International under the category of M-Pharm. Today, I am presenting my research title, Enhanced Dissolution and In-vitro Release of a Multi-Unit Pellet System Tablet for Hypertension Management. This work is based on the theme of in-vitro release and permeation study of conventional and novel dosage form. Hypertension. Hypertension is commonly known as high blood pressure, the condition in which the force of blood against the artery wall is considered too high. Normal blood pressure is around 120 mmHg to 80 mmHg. Hypertension affects over the 1.28 billion people globally. Current oral medication facing the issue like fluctuation, dosing, and poor patient compliance. Multi-unit pellet system offer distinct advantages such as control and predictable gastric empty, reducing the dose dumping, improving the viability and patient compliance. The present work focus on development a bilayer multi-unit pellet system tablet combined of floating sustained release pellet of drug A for prolonged action and immediate release pellet of drug B for rapid onset of action. Aim and objective: formulation, evaluation, and optimization of drug A sustained release and drug B immediate release multi-unit pellet system bilayer tablet for hypertension management. Formulation aim to enhance the dissolution, optimization in vitro release, and providing a more effective and patient-friendly treatment approach for hypertension management. Objective: to perform a pre-formulation study, to perform an analytical study such as HPLC and UV quantitative analysis, implementation of full factorial design. Formation, development, and characterization and optimization of sustained release floating pellet and immediate release pellet. Formulation, development, and characterization and optimization of multi-unit pellet system, comparative analysis of optimized batch with marketed formulation to conduct the stability study as per as ICH Q1 R2 guideline. Rationale of this research is complementary action as the combination of ACE inhibitor and diuretic give a synergistic effect to treat the hypertension, optimization of drug absorption, reduce the side effect, and enhance the safety. As we know, the drug A start degrade in alkaline pH. Therefore, gastroretentive drug delivery system is used to prolong its retention in stomach and ensure the optimal absorption and stability. Morning synergic phenomena, the urge and gradually release of drug helps to manage the early morning blood pressure peak, which associate with increase the risk of cardiovascular disease like heart attack and stroke. Sustain an immediate action to control the blood pressure and reduce the first part mechanism. Drug profile as the shown in table 1 for drug profile of drug A and drug B as given below. Pre-formulation study, melting point determination was done by capillary fuse method, in which the melting point was found to be 105 of drug A and 269 for drug B. FTIR spectral analysis of drug A and drug B confirm the drug purity and compatibility as shown in table, functional group and its wave number. Differential scanning colorimetry of drug A and drug B. As per as literature, melting point of drug A was 105 to 107. We get 108 of drug A and 270 of drug B. UV spectra of drug A and drug B in 0.1 normal HCl and water for determining the lambda max. This was essential to choose the correct media and analytical method. As shown in figure, UV spectra and its calibration curve in 0.1 normal HCl and water. Drug excipient compatibility study. As shown in table, ensure the no chemical interaction between drug and excipient by FTIR. This confirmed the formulation physical and chemically stable prior to the process. Development of simultaneous estimation method by UV to detect the release of drug A and drug B in dissolution. Saturation solubility study was done in water, ethanol, methanol, and 0.1 normal HCl. The drug A possesses the maximum solubility in 0.1 normal HCl, that is 7.236 mg by ml, and for drug B possesses the solubility in ethanol, that is 5.67 mg by ml. Formulation process: preparation of drug A floating pellet and drug B immediate release pellet, which both on compressed into a tablet, dissolution in gastric fluid, in which floating layer get float for 12 hours in gastric fluid. Immediate release get dispersed in gastric fluid and give onset of action by entering in systemic circulation. Manufacturing process of extrusion speronization consists of dry mixing of excipient and drug, which on adding the liquid like water and IPA to get a wet mask, which on extrusion and which on further speronization give a round shape by drying. In this table 5, critical process parameter for extrusion speronization is wet mass, extrusion, speronization, and dry. Formula table for preparation of drug A loaded floating pellet. We have developed a 16 batch by taking a different excipient and its in different concentration. Formula table for drug B immediate release consists of sodium starch glycolate as the super disintegrating, taking a different ratio. Formula table for preparation of drug A loaded floating layer and drug B loaded immediate release layer. Flow property of pellet was done by bulk density, tap density, percent compressibility, Hertzner ratio, and angle of repose, which we get the remark of drug A and drug B. Both was good. Characterization of pellet was done by average particle size was done by optical microscope, a drug content, floating time, lag time, disintegration time as shown in the table. In vitro release study to evaluate the drug release profile for formulated pellets and multi-unit pellet system was done by USP type 2 DISO apparatus in 0.1 normal HCl method. Overview is given below, in which drug A SR layer was done up to 12 hours, and for immediate layer it was done for 60 minutes. In vitro dissolution study for drug A loaded SR pellet consists of 16 batch, in which batch number 16 was optimized batch, possessed the maximum drug release of 42.26% release in 8 hours. And for the immediate release, batch 3 was the optimized batch as the possessed the maximum release of 91.2% release in 40 minutes. Optimization using the full factorial design consists of independent variable HPMC K100M and sodium bicarbonate, taking in lower limit and higher limit. Depending variable was IVRT of drug A and floating time of MUPS. As shown in figure, in vivo ANOVA for quadratic model of both the IVRT and floating time. Preparation of multi-unit pellet system, having the several advantage comparing the conventional, reducing the variability, improve the release control, improve the viability, therapeutic effect, increase the patient compliance. Characterization of multi-unit pellet system done: diameter, thickness, hardness, drug content, weight variability, lag time, floating time, and disintegration time. In vivo release for the MUPS was multi-unit pellet system was batch number 3 was the optimized batch, in which in vitro dissolution profile was found to be 95.57% in 12 hours, and for the immediate batch number 3 get the 95.7% release in 60 minutes. Swelling index for optimized formulation. A high swelling index indicate the tablet has a greater tendency to absorb water and expand, which can affect its floating ability. At the optimized batch, that was F3, since the swelling index was found to be 0.325 in 30 minutes, and in 6 hours we get 4.039. Comparative analysis of optimized batch versus marketed batch. The in vitro release profile of optimized batch F3 of drug A and its marketed product show the distinct difference. The batch number 3 for floating sustained release MUPS release was 95.75 for drug A over the 12 hours, while the marketed product achieved 96.39 release in 30 minutes. Similarly, optimized batch for F3 for drug B immediate release MUPS release was 97.76 in 60 minutes, whereas the marketed product reached 101 in 20 minutes, indicate the faster release. Drug release kinetics for the sustained release follow the Hexan Crown club route, and for immediate it follow the zero order release kinetic. Stability study for optimized batch was done according to ICH Q1 R2 guideline. Till now, 1 month stability was done. Was done. Lag time determination. Lag time of optimized batch F3 was found to be 22 seconds, as shown in the video. Lag time. Floating time determination. In vitro dissolution profile for the optimized batch was found to be 95.55 in 12 hours, and floating time was up to more than 12 hours for drug A, whereas drug B was found to be 95.76 in 60 minutes, in which we show that a drug A, which floats on the upper layer, and the drug B, which is in bottom, it gives the onset of action. RP-HPLC annihilation method development. As shown in table, it is the optimized condition for HPLC. HPLC method validation linearity as shown in the figure A, B, and C. It is the validation method by linearity, accuracy, method precision, and system precision as shown in the figure. Analytical method validation for HPLC chromatograph, as the validation parameter was found, wavelength was 226 nanometer, retention time for drug A was 2.71 minute, and for drug B was 3.129 minute. Linearity range, correlation coefficient, precision, robustness, and accuracy was done and shown in the table. Force degradation study. Conclusion: in the present study, drug A and drug B was selected as the drug molecule performing multi-unit pellet system to manage the hypertension, in which the 16 batch for drug A floating layer pellet was prepared by taking a different concentration of excipient, in which batch number 16 was the optimized, and for drug B immediate release, batch number 3 was optimized batch, which gave 91.2% release in 60 minutes. For the multi-unit pellet system, 9 batches was prepared according to the optimization batch, which consists of different concentration of HPMC K100 and Udrazit RLPO, in which batch number 3 was the optimized batch. The process multi-unit pellet system formulation consists of 30 mg of drug A and 12.5 mg of drug B. Conclude to be promising approach for the managing hypertension. However, the further hypertension activity was required in order to justify its proof. Reference. Thank you.